Good morning, everyone. This is Dr. Pete Delanoy. Usually in the past, I've gone by just Dr. Pete, and today is the very first episode of a new project of mine. I am the founder of an organization I call the Origins of Human Metabolic Disease. And right now, I am an organization of one. That is me, and I'm doing all of the things that my organization has the mission to do, and I'll get to that in a second. Let's start by just letting you know that we are going to talk about some of the fundamental science behind chronic metabolic diseases today. And this journey of mine started 8 years ago. And part part of talking about the science requires that I also talk about my story, because you got to understand how I got from where I started to where I am now. Because back in the day, I started a YouTube channel and I was among the millions. I think you could say that there's that many. It's hard for me to gauge how many metabolic influencers are out there now. Just about anybody that uh, is concerned about their health opens a YouTube channel and they're talking about what they did to, either achieve better health or reverse or at least get into remission their chronic metabolic disease, or at least improve their health. And in the beginning, I started the YouTube channel, and this video is posted there live right now, Dr. Pete's Keto Club, and I was doing the same thing, talking about menus and recipes, and talking about the fact that I presented data from Virta Health and whatnot, how I utilized their randomized controlled trials. There was a number of publications to reverse my own pre-diabetes. And then eventually I talked about the biochemistry behind gout and put my own gout into remission. Now that 8 years of time frame bringing me to today has changed really in what it is I want to do and what I want to accomplish uh, as a scientist who is deeply concerned about our metabolic condition around the world, predominantly here in the US, but whatever happens here happens around the rest of the world too. And so I want to talk about that a little bit today, in addition to some of the science that I'm going to address. I'm also utilizing a new format because, in addition to being a trained PhD biochemist, I was a teacher for 28 years, and I've taught complex chemical concepts to all age groups. I was a university professor for a decade. I've taught at the secondary level, and I finished the last 15 years of my teaching career in a top-rated charter school, a world-rated charter school that had grades from 6th through 12th grade, and I was a straddler. So I was teaching college-level chemistry to 7th and 8th graders, and then upper-level classes in the high school. A very different kind of program from what you see in most standard high schools in the United States. I have, in my efforts to understand chronic metabolic disease, I've married these two parts of my life, that of the research scientist and that of the teacher. And the origins of human metabolic disease, one of the main mission parameters of this new project is to educate people about the fundamental biochemistry that's driving these diseases. What can we look at in the published data, extending back as far as it takes, whatever, and looking at well-done biochemical and molecular studies, paper by paper, in peer-reviewed journals, in order to synthesize large numbers of publications into valid, well-documented hypotheses that can explain chronic metabolic disease progression. So let's get started. So first off, I was 60 years old when I was diagnosed with prediabetes, and I was a gout sufferer 3 years prior to that. So about 57 when I was diagnosed with gout. And like everyone that I know in this story, a lot of the influencers that I've actually trained with back when I was doing certifications and things like that for health coaching, I'm not doing much health coaching anymore because I have shifted all of my energy into studying the science, proposing well-documented hypotheses that can explain our condition. How did I get sick? How did I get into this condition? And then based on well-documented science, what should be the nutritional platform in order to reverse these metabolic diseases? And I'm talking about everything from type 2 diabetes, chronic kidney disease, um, coronary artery diseases, and so on, right? So I'm talking about the full gamut here because if you take the time to look at science, and I think that any well-educated PhD in this area that went to a really good research institution for their PhD understands the fact that when we start thinking about how you get from point A to point B, right? So when we talk about a, a group of people, and some of them are suffering from diabetes, and some of them from obesity, and some of them from cardiovascular disease, most of them from multiple comorbidities. So somebody who's diabetic, uh, there's a high probability that they're also significantly overweight or obese, and there's a significant probability that they have cardiovascular issues on top of kidney issues, right? Especially if they've been diabetic for a really long time. I mean, many, many years down the road, 10, 20, 30 years. Somebody like that is going to have multifactorial issues going on with their organ systems, maybe even including dementia and Alzheimer's. And one of the things that really stands out when someone like me sits back and you look at this is the realization that these metabolic diseases can't have individual reasons why they're happening, right? And they can't have multiple explanations. There has to be central biochemical pathways that drive these pathways. And then when you look at or drive these disease states, and then when you look at the individual genetics, then it becomes apparent why people tend to break out into certain overlapping comorbidities. We're only looking at a few central pathways here. Today, I'm going to be talking about what I believe is one of those central pathways, not the only one. I'm not one of those guys that's going to have a light bulb go off and go, oh, well, because I think when we look at the whole realm of data for what I'm going to be talking about today, you realize that, okay, this is definitely a central pathway that's driving chronic metabolic diseases, every single one of them that I can think of, including, and this shift recently in the low carb space to really like start to talk about mental illnesses and things like that. Yes, even the drivers of that, because this metabolism is in the fundamental housekeeping cells of the brain that run the neurons. So like this is a systemic thing. And as a biochemist, you realize a couple of things about this if you're worth your salt. First off, we're talking about a minimum number of central pathways. And those pathways need to be able to explain the origin of all of these uh, different diseases, right? I hesitate even to call them different. I think they're basically just different ramifications of the same general biochemical pressure that's being exerted within the individual. And that's a really important thing to think about because our biochemical pathways date back more than 3.5 billion years ago. And 3.5 billion years ago, when these things started to come into being, I'm talking about the glycolytic reactions and other related biochemical types of reactions, we didn't have macroscopic control mechanisms around like insulin and cortisol and those kinds of regulatory points of within our metabolism, even though on a large scale now, whether you look in the medical establishment or you look at all these influencers out there that are, that every day you can click on a video on YouTube and you hear all about the fact that the low carb guys don't know what they're talking about, and that it's just calories in, calories out, and that you got to have elevated glucose coming in by whatever source it's going to be in order to exercise and all this kind of bit. Um, and and then we got even in my space, the low carb guys going, well, the low carb is the perfect human diet. And I think for any serious biochemist who looks at this, the very first question you've got to ask is, how did I get sick in the first place? That's what I asked myself 8 years ago. And because that is the key question when you find yourself actually sick, right? So maybe you're this obese person and you can't stop eating sugar. Same difference. The first thing that happens is you stand back and you go, well, I think a reasonable individual steps back and goes, okay, how did I get here? Why am I sick? And when you go to and so you sit down with your doctor, you hear from the doctor, well, you have a metabolic illness, and there's no ultimate treatment for this. There's no cure for it, but we have a lot of really amazing medications that you can take, and we can slow the progression down, right? We've all heard that. But the fact that when that conversation happened with me and my doctor, the doctor uttered the word metabolism. He was using the word metabolism to a PhD biochemist. And I realized, in fact, I might not even have heard the rest of the conversation really. I took the part away about the good meds and the slowing the progression from the email that followed up that meeting. That word metabolism stuck in my mind because I recognized even back then, okay, if this is metabolism, this has to do with what I'm putting in my mouth. Now, that's the short story, because the truth of the matter is metabolism matters for everything. I don't care whether you internalize a toxin through your lungs. The way that toxin is going to get dealt with is going to be metabolism. And the reason that I'm bringing this up is because it may sound like a tangent, but it's not. It's the reason why all of these other factors, if you're a health coach out there, you're talking to your people about getting appropriate sleep, the reduction in stress, the elevation of the amount of exercise that you're getting, and so on, and the types of exercises that you should be doing over time in order to be a healthy human. Why does all this stuff matter? Because it all goes back to metabolism. There is absolutely no way to escape it. And the metabolism is ramped up every single day once you get out of bed and you start putting things in your mouth, right? There are different pathways that are going to be opened and closed, and there's going to be alterations in gene expression that's going on while all of this is happening. And so it matters, perhaps, as we've learned in a lot of different certification classes and things like that, that it that metabolism, what you put in your mouth, or the diet that you have chronically every single day, is perhaps the majority of what it takes to get healthy. Making the choices about what you put in your mouth matters perhaps the most. But truth of the matter is, as we lose this weight and we become leaner, and we lose that excess body fat, these other factors ramp up in their magnitude of importance. That is an important concept here. And the reason that I can say that is because I've been low carb now for 8 years, and I've dealt with every permutation that I hear, especially from some of my most significant mentors, for example. Some of these not having nothing to do whatever with low carb, but come into this from the area of physical therapy, um, and the exercise fitness part of the world who advocate consistently in every single day that you must eat the standard American diet, that you must have the diversity of that food pyramid. You've got to be having, and plant-based the better, right? I've listened to that until I'm blue in the face. But on the other side of that coin, I also hear, or there's an emphasis in my own space that I feel like is also equally unhealthy. And I am going to get to that today, whether or not I maybe have already lost some of the people here. So let me just say right away that when I was first diagnosed as being pre-diabetic and I was a gout sufferer, uh, the first question I had that popped out was, how did I get here? And if this has to do with metabolism, then I need to get into the literature and start to look at this. And from a hardcore biochemical point of view. And of course, the first papers that I got to were the 1 and 2 year studies that Verta Health had already published in the literature. And I actually used those studies to design my lifestyle, which I ended up turning around my prediabetes very rapidly. It was only in a few months that I was able to completely alter my fasting glucose and my A1C. And I don't know if I'm going to come back to that in today's presentation or not, but there are some things to be said about that too. And this has to do with the choice of very limited number of biomarkers when you're trying to make these decisions about, how you should eat um, and your exercise and other issues. All right. But moving on. All right. The other thing, and this was the secondary question at the time that I was asking. And I'm going to ask this. You can read it. It's right in front of you, but I'm going to verbally state it. And at that point, I'm just going to tell you that up until recently, over the last year or so, I was doing what everyone's been trained to do as an influencer, right? I was publishing sometimes upwards of 3 YouTube videos a week, and I was showing up in every electronic forum that you can imagine. And when I looked at the biochemistry of sugar addiction, because again, here's what I want to point out to you. All these diseases, we can make a list of them. They all come from a central mechanism or a combination, including branch points of central mechanisms. You should be able to track the need that somebody has to put sugar in their mouth back to the basic biochemistry and the signaling that's coming or happening at an intracellular level that extends to an extracellular level, that then those signals are passed on, however they get there, through our nervous system, for example, into the brain, where you have the activation of more metabolism, and the expression of certain proteins that turn around and drive the motivation that person has to go sit down and have more of what it is that they're eating. Yes, those pathways exist, but when I went into some of these forums and I said, okay, let's just ask the question, is sugar addiction real? Oh my God, the backslap that I got was not only excruciating, but it was also really disheartening. And it's because that these individuals don't understand why I'm asking these questions. It's not because I doubt this stuff, right? Because I've looked at the science and I know it's real. The reason why I ask the question is that you, as a scientist, you must constantly refine your hypotheses and your theories about this. And that means asking a similar or same question over and over and over again. Science is constantly changing. The data that we get, at least in this area of nutrition and the reversal of chronic metabolic diseases, is only getting better. The more of it we have, doesn't it doesn't uh, discriminate what we already think about it, it makes the argument stronger. So I'm asking the question, what is the optimal human diet? Now, I know in my own space, there's a lot of people that already, a lot of influencers, very, very famous ones, that think they already know the answer to that. And today we're going to talk about the science, which they think allows them to make that assertion. And I am not going to make an argument against the science that they have, but I want to talk to you guys directly about the difference in the science that they're looking at, the perspective that it provides versus the science that I look at and the perspective that it provides, because these things are really in two different places, right? We're in one case, you're looking from the outside in in a very complex way at the human population, which is a lot like looking at a forest. And then you recognize that in the forest, especially the trees that you're looking at, a distribution of them, that the majority of those trees are not doing well. And you're trying to figure out, they're not doing well metabolically, and you're trying to figure out, all right, what's wrong with them? So we start doing these observational studies, we start doing these clinical studies. I'm going to talk about one in particular near the end of my presentation that everyone is going to be aware of at this point. And we are looking for solutions to a problem that we cannot find without going inside the forest to the center of the issue, which is inside the cell. It is at the level of our biochemical pathways because they evolved 3.5 billion years ago. And not long after their evolution, the microscopic control of those pathways evolved. And that was a long time before we ever got to cells and to multicellular organisms and beyond that, where these macroscopic control points became so important to our existence. I'm talking about uh, the one thing that we all understand together, I think, in this space especially, is insulin and insulin resistance. That's a macroscopic control, right? I dwell underneath that, and I'm looking underneath that for answers to these crucial problems that face our population right now. And I can offer some solutions. I'll get to some of that. So underneath this question of what is the optimal human diet, what does our biochemistry have to say about this? Now, one of the things that I've been accused of a lot since I started doing this stuff, especially in the days when I was publishing videos that offered the science. I was in the weeds. I am in the weeds. And you know what? We have to go into the weeds. We don't have to like it, but we have to do it. And the reason that we have to do it is because we need to understand how our biochemical pathways are actually functioning. And when we step back from this and we listen to a lot of the, like I was at COSI midwinter sometime around there, and one of the speakers was Nina Teicholz, and everyone knows she's working on trying to get the food pyramid redone so it actually reflects science and gives people a better idea what they ought to be eating for their health and so on. And we all know this story. And any of the guys out there that have trained to be health coaches and got their certifications, we've talked about the time frame when the nutritional guidelines were published. They came out in I think it was in 1983, but the process to get there started well in advance of that. And the purpose of this video is not to go back over that history. All right, I don't want to spend my time on that. I'm just bringing it up because you guys can relate to it. And here's the thing, though, that no one talks about. It was in the same time frame where the funding at NIH was shifted away from funding classical biochemical studies. I'm talking about the type that Krebs did, and perhaps the most I'm going to use the word infamous, because this guy deserved so much more credit than he's given today, and that's Richard L. Veach. At the time when their funding was cut off, they had discerned and had published actually extensively about this microscopic control of our fundamental metabolic pathways. You hardly see any discussion about these concepts in any university level biochemistry textbook. Everything there is taught as settled science, as if we know that glycolysis is the only way that glucose is processed, unless you're talking about the synthesis of glucose, of course, which comes from gluconeogenesis. Either way, we talk about these individual pathways, like, for example, the synthesis of fat, right? And you can look up in any textbook and you can find the chemical, the biochemical steps for de novo lipogenesis, but nowhere in those textbooks do you find a discussion how these biochemical pathways communicate with one another and how they shift using parts of sequences, pasting together these parts of sequences to do certain functions, and how those fluctuations are happening. And in our space, I think the best example of this is comparing glycolysis and gluconeogenesis, because those two pathways are happening over the same sequence of chemical reactions. And if you just step back from that and you think about it for a minute, then you realize that that can't happen, right? You can't have both of those pathways operating simultaneously. It would be like having a one-lane street for glycolysis going in one direction, and then the realization that glycolysis also goes up that street, and that if they're both going at the same time, there's going to be a car wreck. That's kind of what you have going on there. It's a little bit more complex than that, and I'm not going to get into it now, but you basically can only flux in one way, or you can flux in the other. So how is that level of control happening? And I need you to be really careful here, because a knee-jerk reaction to what I just said is to go, well, there's a difference between glucagon and insulin, Dr. Pete. That's the answer. No, it's not. Are those two macroscopic hormones involved in this transition? Yeah, of course they are. I'm not saying they aren't, but I can tell you right now, there's microscopic control that's involved in this, and it happens first before this downstream set of macroscopic controls that are jumping in, or it happens simultaneously with it, right? But it's something that we don't talk about or we don't hear about, and that level of science, essentially, it's there in the literature for those that want to go and read it. It's super complex and super difficult to understand because it involves the areas of thermodynamics and kinetics, and biochemical kinetics and biochemical thermodynamics. And all you need to do in a classroom, I don't care whether it's at the high school level or at the university level, is raise the issue of thermodynamics, and just about everybody in the room is going to run out of the room screaming and waving their arms. Oh my God, don't go there. I can't understand it. I won't understand it. I don't want a damn thing to do with it. And I'm going to tell you right now, we've ignored microscopic control of these biochemical pathways for too long. We need to discern what these pathways are. And there's a lot of really good, high quality science that's out there. And I'm going to talk about one of the fundamental pathways that I talked about in the past in my videos, and then recently in a collaboration with a number of really good scientists, including Rick Johnson. We published a comprehensive review that is really the first step in defining this entire pathway, how fat is synthesized, how glycogen and fat is synthesized in the liver, the first concerted pathway with all the connections, right? This is a very important distinction. But that work is also ongoing, and there are more publications in the pipeline. So let's move on to now the next slide. Okay, this is just announcing that I have started this organization. I am an organization right now of one scientist. You can email me if you have questions after this presentation at the email address I have here. So you can look it back up in the video that will be posted. It will be here on LinkedIn. It will also be posted on my YouTube channel, Dr. Pete's Keto Club. I'm still utilizing that channel that way. And then for those of you that see the validity in the science and you think that someone like me is worth supporting, you can go to my Patreon, and that's where I have all of the deliverables that were ever created for all of the health coaching that I did. This includes support materials for reversing diabetes and obesity and cardiovascular, the improvement of cardiovascular condition, and putting gout in remission. It's at my Patreon here. It's $10 a month. You literally can go on there for $10, download everything that you want. There's also an online course there that's actually a really good course. It's only a couple hours long that explains how to do the low carb lifestyle. You can get all that information and then just cancel uh, the Patreon membership. Soon things will get a little bit more sophisticated with that. So this is how you can get a hold of me. This is what I'm doing. Eventually I'll be a nonprofit, and hopefully there will be more of me's that are involved in this. We have a just cause, and my just cause is to build a world where the majority of people wake up metabolically healthy, physically robust, mentally resilient, and inspired to live a long life. This is a world problem, although in westernized communities, it is greater at this point than it is uh, uh, anywhere else. The mission, though, is to fill a space that right now in our low carb space, or in the medical community, or in the general human population here in the US, we are not focused on the instrumental biochemical pathways that drive metabolic diseases. And my main mission, which is what I'm working on full time right now, which is why you guys don't hear from me very often, because I am literally writing manuscripts every single day. I'm working on a manuscript, and I'm doing that now, and it's something I hopefully can get off my plate in the next couple of weeks. And then yet there will be another one, because the main mission is to discern how these pathways actually work. Once we understand, we have a well-documented hypothesis, then the priority of my organization is to inspire the shift of grant money back into classical metabolic studies, and to find research scientists that are willing to take on this mantle and go forward with actually defining the holes in these hypotheses. They are hypotheses, and that means they're going to change. And that means there are problems in the hypotheses, questions rather, that need to be answered. And then the other aspect of my mission uh, is to enhance what the practitioners know about the nuances of what needs to be done for somebody who is suffering from a particular metabolic disease, the best way to go forward to get remission, or I mean, ultimately what you would like is to be able to get reversal. And anyone who's been in this field as long as I have, actually now, I'm in my 8th season of it, and I have extensive health coaching experience at this point, 7 years, with people all around the world, and also significantly here in the US, that people are individuals, right? And finding a solution for an individual that may have a different overlap of chronic metabolic diseases is not necessarily the same as somebody else who's suffering from a different set. You have to take the individual. When we understand these biochemical pathways, and we know how the microscopic region can drive, for example, insulin resistance, then we can make better choices about how we structure the diets that they're on, the exercise that they're doing, how, all of the different factors that feed into our metabolism. So I just briefly want to talk about the science a little bit. I'm going to draw your attention to the right. This is just a screenshot of our recent publication. This is a benchmark publication because it's the first publication by anyone that has fully synthesized the data of glucose, fructose, uric acid metabolism to show that, or to make the argument in a well-documented fashion, that this is more of a general process than either my own community, the low-carb space, or the medical establishment. Everyone else has taken this thing to be. When most people listen to Richard Johnson talk about fructose, at the end of the conversation, they go, well, I eliminated fructose. I don't eat sugar, right? I could go off on a tangent about that comment, but let's not do it, right? And therefore, I don't have this issue. Well, it turns out that you can make a really well-documented argument going back decades on the enzyme aldose reductase to show that fructose metabolism is way more general than you think. And you might even be more correct to call this. And in fact, I'm going to call it now that it's glucose, fructose, uric acid metabolism. This pathway is, in many respects, maybe it's more dependent on glucose because of the fact that most of the fructose that you bring in through your mouth is going to be converted to glucose in the intestine. So what the pancreas and the liver actually see is an elevated level of glucose, in the sense that not all the glucose is coming directly from the meal. A large portion of that glucose, if somebody ate sugar with the meal, is coming from fructose that was converted to glucose. And when the glucose concentration that enters into the liver becomes high enough, which isn't that high, people, by the way, when you look at the enzymes that are receiving that glucose, they actually have a pretty low threshold where they become activated. And I don't want to use a number here because hopefully I'm talking to a wide variety of different people, not just scientists. But so suffice to say that this threshold level of glucose that has to rise above that is actually a pretty low number. And when that happens, the polyol pathway is activated. We produce endogenous fructose. This is not only documented, not in this paper in particular, right? This is another manuscript, but polyol pathway is documented in human tissues everywhere where it's been looked for, right? So off the top of my head, that means pancreas, liver, adipose, muscle, kidney, brain, right? And also in the chondrocyte. So this is a very important pathway, and it, in all likelihood, is ubiquitous. And that means that whether or not we activate glucose fructose metabolism doesn't depend on exogenous fructose. Hyperglycemia does the trick, along with, there are other activators of this pathway. And I don't want to go into the weeds on this pathway right now. This is not really the purpose of this video. What is important to say about this is that this is a central pathway, and I'm going to get to the reasons why in just 1 second. I want to point to the left to the chemical aspect of this, and don't worry, I'm not going to go through this. This has been one of the big complaints that I get over the years, and probably the main reason I don't get invited to a lot of the low carb talks is because everyone's afraid I'm going to do this. I'm going to talk about the science. The reason I'm showing you this is because you need to know that this is really the first blow-by-blow concerted pathway that's been shown for the synthesis of fat in the liver. This is all previously published data that is known science. And why hasn't someone talked about this before? Because nobody's looking. That's why. All right. And this beautifully shows a couple of things. One, how glucose is working in this situation. It also proposes things that come literally out of Veach's work and his suggestions of microscopic control. You can see it there in the relationship between the chemical reaction that's producing lactate and the reaction that is catalyzed by isocitrate dehydrogenase 1. And guess what? Glutamine metabolism is involved in this. Who would have thought? Now, if you follow cancer, you realize that Thomas Seyfried is out there. I feel akin to him because he is somebody who's doing what I'm doing, but he's doing it in cancer. And he's been doing it for a lot longer than I have, right? This guy has a whole career wrapped into this. And unlike me, he has a laboratory. So he's done a lot of these studies himself, right? And he has a pedigree, a lot like Johnson, hundreds and hundreds of papers that you can go into and you can walk through. And you should listen to some of his stuff. And I'm going to get to some of this today, the importance of this, because the metabolism that I am trying to educate people about is sitting underneath the main concept he talks about, which is the generation of mitochondrial insufficiency that leads to systemic pressure for cancer, right? This is an important thing. So on the left, you have the whole pathway there. We're in the process of publishing more papers on this, but our paper on the right introduces this model, right? Okay, so what's the deal with activating glucose, fructose, uric acid metabolism? Well, this is it. This is a primary pathway for the synthesis of glycogen in the liver, for the synthesis of fat. Where does this fat thing come in? Driving non-alcoholic fatty liver disease, or in today's new acronym, MASLD, MASLD. But the thing you got to remember is, if this is the first time you've heard it, then it's not a question of remembering it. But when we talk about producing fat in the liver, we're talking about pushing triglycerides into the bloodstream and also VLDL. And where does this have an impact? Think about it. Lipid energy model. And I'm going to also talk about the fact that when we activate this pathway, we drive hyperinsulinemia. We drive insulin resistance in the peripheral tissues. We drive hyperglycemia. Where does all of this come into play? Right? In the peripheral tissues. This has to do with also driving the carbohydrate insulin model of obesity. Do you see how central mechanism, going back into our biochemistry, can start to explain all of these on the surface disparate theories we have about what's going on? And then turning around and going, well, if we go low carb, we can reverse those things. Why can we reverse these things? Well, when you reduce the input of sugar, when you reduce the input of alcohol, when you reduce the input of hyperglycemia during mealtime, and you go to a low carb environment, guess which pathway you essentially downregulate? Not only downregulate, but you put it in a situation where it can't open. Ba-ba, this one. All right. Now, perhaps most importantly at the bottom in red, when we activate this pathway on a natural level, right, in evolving humans, this was a good thing because we only ate this way intermittently. And we needed to be responsive. The survival of humans depended on our ability to respond to certain macronutrients and build fat quickly and efficiently, restore glycogen, but then also these other aspects. The systemic inflammation is about stimulating the immune system, right? Which we needed to do. The alteration in nitric oxide signaling downstream, this has the effect of altering blood pressure. And you want, when you're eating this way, you want the blood pressure as a human that needs to survive. You need the blood pressure to go up in order to get nutrients out to all the external tissues as far as they can go. So at the bottom of this, we see in red that when we activate this pathway, we alter mitochondrial function. This is important. Okay, this slide on the right is to emphasize the fact that this is systemic. So what happens in the liver doesn't stay in the liver, right? There's extensive signaling. I talked about it a second ago, the hyperinsulinemia, hyperglycemia, insulin resistance, all of that stuff, all of that signaling is propagated downstream throughout our organs, even including the chondrocyte, which is down there on the left. That yellow tissue over there on the far left side of that illustration are adipocytes. You see the brain in there, muscle tissue, kidneys, all the primary organs are here, and they're all responsive to the signaling that gets propagated out of the liver when people are eating basically in a way that activates this pathway. And then very importantly, here on the left, I'm talking about this concept that I introduced a second ago when I was talking about Thomas Seyfried, who is the scientist that is trying to get the world to listen about the fact that cancer is metabolic. And here you can see how glucose, fructose, uric acid metabolism, it drives a change in mitochondrial function. But when somebody's eating this way and they're doing it chronically from the time they get out of bed until they go to bed at night, literally with a dessert just before bed, most likely, this change in mitochondrial function eventually becomes chronic and you get mitochondrial damage. You can actually, when you look at these papers, that you can look at pictures of the mitochondria and you can see that they've changed their structure and their form and whatnot. They are not functioning normally anymore, and this leads to mitochondrial insufficiency, which is at the foundation of all cancers, quite frankly. And this is why Seyfried, and he's published hundreds of videos about this, I think it's hundreds at this point, talks about the fact that you can utilize the ketogenic diet, right, a proper one for reversing or putting cancer in remission, coupled with glutamine manipulation, and I think they call it press pulse, and combining the modulation of glutamine metabolism with a ketogenic diet, you can transform the life of somebody who's suffering from cancer, and that's all cancers, right? So how do we get to the mitochondrial insufficiency? This is a central pathway, what we're talking about today: glucose, fructose, uric acid metabolism. There may be other pathways, right? But this is the central one. Now I want to talk about our community a little bit. Here we're looking at some statistics, and you can, we've all seen these, and hopefully I've got some people listening that are not in my hallway, right? That are sitting, looking at this from the outside in would be really beautiful if that's happening. But we have these statistics, 29 million people approximately, right? It depends on which organization you take these stats from. 29 million people suffering from type 2 diabetes. About 98 million are pre-diabetic. That's where I was. About 40% to 50%, depending on where you, again, get the statistics from, are obese. And when you factor in the people that are overweight, that's the situation that I was in, right? I think people break out into different body types with this, but I don't want to get into a different tangent. When you look at the overweight combined with obesity, you're looking at somewhere around 74% of our population. It's an amazing amount. We have, I put this in there because I think that gout is one of the most underrated chronic metabolic diseases we have, and it's also a window into the soul of chronic metabolic disease because it is driven by glucose, fructose, uric acid metabolism at the level of the chondrocyte. This is fundamentally a central part of this. And it really was when I started looking at the biochemistry of gout going back into even papers published in the 1850s, uh, it made me realize that I was looking at the central pathway, and that uric acid was, that took me to Rick Johnson, which here I am eight years later, basically. 20% of our population, 52 million, are hyperuricemic. And you know what? Nobody, including me, has a good explanation for that. I have some ideas about it, but I'm not going to get into those here. But here's the thing. Uh, there's a ton of hand waving with this all around. There are some in the medical community that say you need to lower it using uric acid lowering drugs. There are some that say, okay, well, hyperuricemia at 6 mg per deciliter is now the average, and you shouldn't even worry about it. And in the low carb space, you have two sides of that argument too. And I don't know. I'm going to tell you that right now. I don't know what to think about it. And I'm not talking about looking at observational data. I'm looking at real biochemical data, and I'm not sure what I think about it, because we know that uric acid can diffuse into a particular tissue and then elicit the same intracellular effects that it elicits when it's produced directly by that tissue. So that's an open question. That's an open question about the hyperuricemia. It's something that we really should be pursuing significantly more than we are. And in the low carb environment, we have to care about this, because when someone goes ketogenic that is normally hyperuricemic to start with, like me, or they go carnivore, they see this big spike in uric acid in the early days before keto adaptation. And then uniformly, when that uric acid comes back down, and some people are going to argue, some scientists and medical doctors are going to argue, well, you're just coming down to the average uric acid in our population. I would say, okay, wait a minute, because in the early 1900s, the average was around 4 mg per deciliter, not 6. So obviously something has happened in that interim to change things. And that change is not an evolutionary change. There's not enough time for that sort of thing to have happened. So this is an open question, and the reason I have it here is because almost no one ever talks about it. But I do, because it's a real thing. All right. And then finally, chronic kidney disease. This is one of those comorbidities that you find literally in most people that are suffering from diabetes in the long term, right? And also in people, like for those of us that are 60 years old and older, there's a couple things I want to say about this. Almost all of us suffer from some form of chronic metabolic disease. Almost everyone. And from a male point of view, I have to tell you, in my extended family, and then in most of my friends that I socialize with that are my age, they are many of them are suffering from prostate cancer. And again, don't forget about what I talked about with Thomas Seyfried, right? This is a metabolic disease, and it's driven, I guarantee you, by glucose, fructose, uric acid metabolism. So chronic kidney disease is a factor for all of us that are aged and also suffering from. Now, we can see, I'm going to open the subject about this now, that this circle that I drew in the middle here that says US, that's not the United States, that's us, right? That's this population of older people, 60 years old or older, that are suffering from chronic metabolic diseases. And I want to point out that when you go out and you peruse on YouTube, or you just listen to the videos that are being posted on LinkedIn, almost uniformly, everyone is focused on, they call this science, right? What I want to point out to you in this video as I close is that not all science is the same. There's different aspects and different disciplines that are important to try and understand our human biology. What are the factors that drove us to become chronically metabolically ill, right? That's question one. Two, what is the ultimate human diet? How do we answer that question? Well, it can't be answered by virtue of the fact that if you take somebody who's chronically metabolically ill and you put them on a carnivore diet, or you put them on a ketogenic diet, right, that's been individualized for them in both cases, and you see reversal or remission, that is a really important piece of data, right? That says that when we get them away from the standard American diet, we stop letting them pick from that ridiculous food pyramid, right? That we see almost immediately a shift towards improvement. But we also know that if the diet is done right, that we get reversal for sure in diabetes, right? Unless the individual's been type 2 diabetic for 30 or 40 years, those individuals might already have enough permanent kidney, liver, brain issues, right, that are not reversible. But even in those cases, you're going to see their situation improve. You should be able to get remission. You should be able to at least stop the damage from progressing any further by doing that. Now, what's missing at all levels in our society, right? There's a major push for more clinical studies and all that. I'm not arguing against that, right? But I think it was Nina Teicholz in a video or somewhere that I saw the clip. Maybe it was at COSI when I was there this winter at that conference. And she said that there's somewhere over 100 clinical studies that show you can reverse diabetes, right? And I've said this in my past videos. We know this is no longer a hand waving argument. We know we can reverse diabetes utilizing a ketogenic diet. Why? Because we've done it, and 100 studies should be enough, right? I'm not saying we shouldn't do more. We do. But there's this other aspect of science that Krebs, who died in the 80s, very early 80s, I believe that's correct, and his student Richard L. Veach, when they lost the funding, they had clearly identified this level of microscopic control. And there were other guys too, other scientists, Williamson, Atkinson, a guy named Harold. All of these guys wrote about this microscopic level control of our biochemical pathways and how metabolic decisions are being made and how you can link sections of one pathway to another. And that to avoid futile cycles, that would be something like having gluconeogenesis running at the same time as glycolysis, that these choices are being made and driven both at the thermodynamic level and at the kinetic level. And it's that type of science which I'm showing over here. Whoops. Over here. This is the process. We need to bring that back. We hardly have any of it, right? I'm not saying that there aren't metabolic scientists out there that are doing individualized experiments in their own hallway, but we need to reestablish a group of scientists that are thinking about this problem from a systemic point of view, and to bring those classical biochemical studies back that were lost in the early 80s. Funding was cut off, right? There was this major shift in funding away from classical metabolic studies to the genome, the human genome, and to more pharmaceutical directed types of experiments. Take drug A and layer it on top of some type of tissue culture cell and then see if some major pathway was interrupted. Well, here's the thing. When you do experiments like that, the only data that you get at the end of it is whether or not some macroscopic pathway was either closed or it remained open, and whether the drug worked or it didn't. What have you learned about how our system is working? That's the question I have. And I got news for you, not very much, especially if you're layering drugs on tissue culture cells over a pathway that you actually don't really understand fully how it's operating, especially when you get the integration between that pathway and other pathways, which is constantly going on in our cells. This isn't like we're not doing glycolysis 1 minute. Oh, and then we're going to open de novo lipogenesis over here. And then over here, we're going to do some nucleotide biosynthesis. This is all coordinated action. And the needs of all of those systems require taking inputs or outputs from all these different pathways and integrating this. It's amazingly complex. I'm not claiming that it isn't, but we stopped looking at that a long time ago. And then the fundamental disciplines, thermodynamics and chemical kinetics, no one wants to talk about it. Like even when you look at the newest literature, 2024, 2025, you'll see statements in the introductions of those that, oh yeah, when we look back at the research that was originally done by, I'll give you an example, like from Veach's lab, they used unorthodox methods, or they did that work was underappreciated. Holy crap, people. We got to stop doing that. It's not underappreciated and it wasn't unorthodox. We need to get back to this idea that we're going to be setting up in vitro systems, right? There's no reason why that kind of stuff can't be done. And when we had the shift to molecular biology, for example, the molecular biology of DNA, how do you think we learned so much about how DNA is replicated, how you transcribe a gene, how you take the information from the gene and make it into a piece of RNA? That stuff all started with putting individual enzymes and complex enzymes in a test tube, providing substrate, and then analyzing what the requirements were exactly for that process. Yes, in model systems, but how did we get from that understanding to where we are today? That's how we did it. One thing begets another. We go from in vitro to in vivo studies. Then we land in animal studies, right? Rats and mice. I mean, I pull my hair out when I hear somebody in the low carb environment looking at a single paper with no uh, uh, without the motivation to see how that paper fits into a system, right? Saying, well, it's just a rat study, so we don't have to pay attention to it. Okay. So question number one, did that rat study fit into this sequence that I'm talking about here? Did that rat study originate by substantial in vitro and in vivo work that preceded it? Those are things that need to be known. We shouldn't really ever be talking about individual papers. I have to say that. It could get me into trouble with this community, but you know what? I don't really care. We should almost never be looking at single papers. When I see a single paper come out that says something that could influence how the low carb people ought to be eating, or the standard American people, or my next door neighbor who is a fitness professional should be eating, the first thing I go is, what's context? Where did that come from? Does it fit into this? What is the data that came before it? That's what I'm interested in, because that's where the story is, right? And then eventually that leads to well thought out, well done clinical studies, right? That's where the meat of this situation is. Lastly, I'm going to give you an example. Recently, and this was really big news for our community, because uh, any person who has gone through the process like me, right? You started out being chronically metabolically ill on some kind of level. You decided to do something about it. You got to the ketogenic world, or low carb world, or carnivore world, however you got there. And then you did that nutritional, those nutritional parameters, and you saw yourself, you made yourself healthy. And then what happens, right? We get all psyched up and we go get our certifications and we start trying to also stand on the mountaintop, right, and yell and tell people about this and to help our neighboring humans do the same thing that we have done. But one of the central questions about this, and all of us have run into it, is we go to the doctor and we say, well, I reversed my pre-diabetes. That's what I did. And I did it using a ketogenic diet. And this is what happened actually with me. My doctor put up her hand and she said, we're not going to have that discussion here. Number one. And number two, all the fat you're eating is going to kill you because you're going to have a heart attack, right? So this recent study, plaque negates plaque, it's all over YouTube right now. Incredible work that this group did. Dave Feldman and Sotomoto and Norwitz and Matt Budoff, and I know there were others here too. I'm not listing them right now, but this group of researchers did, and they showed that that the LDL and the ApoB did not drive plaque formation, but on the other side of the coin, they saw that for those of us, and I'm one of those guys that had a CAC score before starting the ketogenic diet, right? Had plaque, calcified plaque before starting a ketogenic diet that we still plaqued. And I'm going to read you a quote from one of the interviews that happened with Matt Budoff by Brett Scher. And that quote says that people want the answer. What's causing plaque is kind of an unanswerable question for one cause. So let me point out one of the reasons, or the main reason why I wanted to do this presentation today, is to make the point that we need to bring back the fundamental biochemical science of our metabolic pathways, because this is one of the ways we can actually get an answer to this, right? Glucose, fructose, uric acid metabolism, the analysis that I've been doing for the first time proposes documented mechanisms that show us how we can get sick, how we can get to the diabetes, how we can get to the obesity, how we get to be a gout sufferer, right? All these different illnesses. And guess what? Glucose, fructose, uric acid metabolism is highly activatable in endothelial tissue. If you just go on PubMed, and I don't like this, I'm doing something right now I told you I wouldn't do, but if you just go on PubMed and you run that search, endothelial fructose, there's over 700, well, there's close to 700 publications there, just at a fingertip. Now, before I got to the point where I was publishing, I spent almost 7 years in the literature on fructose uric acid metabolism. And, probably 50% of that stuff that I read, at least a full quarter of it, at least 25% of it, over 700 publications alone out of Richard Johnson's group. Think about that. I have looked at thousands of papers. The amount of papers that show glucose fructose metabolism is highly activatable in endothelial tissue is massive. It's one of the tissue types that have been looked at specifically. Now, here's what I want to point out to you. All of these clinical studies that are being done are standing on the outside of the forest, right? We're looking at these populations. In this case, it was lean mass hyperresponders. We're looking at this population of trees, and we're going, all right, what is the situation with plaquing with them, right? And this is an outside-in analysis. This is really important for us. I'm not detracting from this, but it's a beautiful example of what I'm talking about. And so here's what I'm arguing to you. Brett Scher's statement, which I quoted directly from the video, is absolutely true. You can't get where they want to go just by doing these outside-in experiments, they're going to get a long ways, and there are going to be many years of more work that needs to be done. This is very fundamental work. So understand me when I say that they need to be doing it. Okay, I'm not arguing against that. But the missing piece of this is the other side of this, starting from the inside and moving out. We need endothelial models. And in fact, I started graduate school in the early 80s. And one of the key things that I learned is when you're trying to learn or understand a biochemical process, you rebuilt that process in the test tube. And given the fact that this is 2025, I guarantee you there's a way to rebuild component by component this endothelial system in a test tube and look and see how the plaquing happens. It can be done. But besides that, there ought to be even more basic studies looking more specifically of the relationship between the input of sugar, specific macronutrients in dosage-dependent ways, glucose and fructose, and how that changes, for example, the glycocalyx. The glycocalyx. Up here you see the book by Malcolm, get his name right, Malcolm Kendricks, which I've read a few times. He has proposed a very relevant possible mechanism for how the plaquing process can happen. And it's a brilliant framework that would, if researchers who are interested in solving this problem from the inside out were to look at this, these systems can be developed in the test tube. And then what do? You take them in vivo, right? And these would be potentially where these more complex systems would be built. And then you would monitor plaquing in that environment, for example. And then like in the last slide, you take it into animal models. And then that's where the animal models meet the clinical trials that are coming the other direction. I want to finish here just by saying we have to inspire the fundamental metabolic science that we lost in the same time frame as when the nutritional guidelines were published. We need to listen to more guys that are like me. Please invite me to your conferences and let's talk about glucose, fructose, uric acid metabolism and the influence it has on these other models that we already are relying on in order to make decisions about how we're eating, exercising, sleeping, and whatever else I forgot. All right. Thanks for your time today, those of you that listened, and we'll see you the next time I'm here.